Okay, I think I should start the class and let's other people. I don't know whether other people join or not, but at least we'll start. Uh, Hello everyone. So, today is our week 4 live session, and we'll be starting Introduction to Proteomics. Uh, Before moving ahead, I want to ask if you have any doubt from the previous session. Okay, I'm assuming you don't have any. So, in today's presentation or today's session, we will be covering 2D differential gel electrophoresis. So, we will talk about basically it is an advancement of two-dimensional gel, and we'll talk about the basic data analysis and application of this particular technique, and then I will talk a little about system biology and proteomics, and definitely we're going to do the previous year question paper. So before, so this is just an image which is showing how differential in-gel electrophoresis look like. It is an advancement of what we have studied in two-dimensional electrophoresis. It is basically allowing you to do a multiplex studies. Okay, so you can run one or two or three samples together in one gel, and it save a lot of your time. So before talking about DIGE, that is differential in gel electrophoresis, I want to talk about the challenges we came across while doing 2D gel electrophoresis. What were the challenges and why we have shifted, like, or why there is an advancement of 2D gel electrophoresis, that is differential in gel electrophoresis. So first thing that is very common when we talk about gel electrophoresis is gel to gel variability. Even if you have a same sample, even if you have done in a same scenario, but still you're going to have a different gel when you run the same samples again in 2D gel. So this is basically, okay, there are people who are joining. So let's just see if I can. Okay, so as I've already mentioned that this is DIG is an advancement of this particular technique. So when you see gel to gel variability, and if you cannot reproduce your gel or you cannot reproduce your gel image as it is, that create a lot of confusion when you are trying to reproduce your results. So that is one of the very important thing. And then at the same time, 2D gel give you a limited protein resolution. So basically you may struggle to separate protein with a very similar isoelectric point. So if I talk about when we say 1D, the main limitation was that we cannot separate proteins with the same molecular weight, right? So we shifted to 2D, where we are using isoelectric points, and we are dealing with PI of these proteins, and first separation was on the basis of that, and then followed by the molecular weight based separation. But what happen if you have same isoelectric point? Even in that case you may not and similar molecular weight, like similar like isoelectric point as well as similar molecular weight, then it becomes very difficult for you to resolve these protein. So it will lead to overlapping of spot, and then you will not have an accurate analysis of your samples. So if you have two protein overlapped at the same spot, then you will have an additive effect of both the protein, and you will never understand exact differentiation of or exact expression analysis of these protein. So that is another limitation. Then you have low reproducibility. When I talk about low reproducibility, is basically based on that first point only, that is gel to gel variability is very high. So and not just that, even when you make a gel, because it's in you have your IPG strip, which is definitely going to be constant, but at the same time you're going to make your resolving gel for your 2D second dimension separation, which is on molecular weight. So while making gel you may have variation, While loading sample, you may have variations, and the condition in which you are running your sample. So even if your IPG strips, which is for one-dimensional which is a one-dimensional separation based on TI, there also you're going to have variation because you may have consumed that buffer previously, and or it might not have the same pH that was there when you run it for the first time. So these are the things that one these are the challenges that one faces while doing two-dimensional gel electrophoresis. Despite using fresh buffers, despite trying to reproduce the same condition, you may have these variation based on different reasons, which is gel preparation, sample loading, and other. Coming to detection sensitivity. So when I talk about detection sensitivity, as I've already mentioned, there are some proteins which are highly abundant in your sample. For example, in serum, you will have albumin. So you have to either remove those sample to have a detection of low abundance proteins, and these proteins may be more crucial when we talk about something to take out something to discover from your sample, like biomarkers. These low abundant proteins are very important in that aspect. So it become very difficult for one to have a good sensitivity when we talk about gel electrophoresis, or you in general gel based proteomics. So sometimes sensitivity is low. Then most of the time it is low when you're using 2D gel. But then there are some advancement in the gel based proteomics also. We're going to study that as well. Then coming to labor intensive and time consuming point, that is very much true about two-dimensional gel electrophoresis. It's very protocol intensive technique. It has a lot of steps. You have to do a lot of things before as well as after, and during the process also you have to maintain you have to take many precautions while running your one-dimensional as well as two-dimensional separation. So these are the challenges that is there, because not just your once you're done with your running, once your separation, two-dimensional separation is done, you have to focus on staining as well, and then you have to do image analysis. These are the steps where one have to be very careful, and whenever you're reproducing your result, at that time also you have to follow the same protocol. So that is there, and it is very time consuming because it take approximately 3 days to complete the entire process, and sometime more depending upon what kind of staining you have done. So that is there. And then you have difficulty in analyzing hydrophobic proteins, because hydrophobic proteins or membrane proteins often exhibit poor solubility in the buffers. They are less soluble, so sometimes even if you have good loading protein dye preparation, properly you have made your lysate, if your hydrophobic proteins are not soluble in your buffer that you're using, that is also creating a lot of problem when we talk about gel-based proteomics. So gel-based proteomics, and even if we come to 2D gel proteomics, they have the similar sort of challenges, because it becomes difficult to run these proteins on your gel. So that is there. So these are the challenges, and then with these challenges in mind, there are some additional modifications done in doing gel-based proteomics, and one of them is differential in gel electrophoresis, which is DIGE. I'll be using DIGE term many times during this session, so I hope you all are aware now that what exactly DIGE is, at least the full form. Then talking about what exactly this technique is about. So it is a variant of gel electrophoresis, or you can say 2D gel electrophoresis, and it allows the simultaneous separation of 3 samples on one gel, bringing a new level of statistical confidence and reliability to 2D gel electrophoresis. So as I have already said that there is a lot of variation, low reproducibility, gel to gel variability in your 2D gel electrophoresis. But when we shift to DIGE, we are running these sample at up to three sample in one gel, and that gives you a lot of information as well as reliability of that particular gel, and I'll talk about how. So we use fluorescent protein labeling dye, which are cyanin dye. They are cyanin 2, cyanin 3, cyanin 5. These are the different uh, color based variation or name of the dye, and they have different excitation and emission wavelength. So when you are studying a particular dye like CY2, then you are using yellow. That will give you a yellow based detection, yellow color detection. Your spot's going to be appear in yellow color. Likewise for CY3 it's red, and for CY5 it's somehow bluish color. So when you have a combined effect of these dye, then you definitely have an overlapped image, and that will give you altogether a different color. So on the basis of that, you study this particular technique. And if I talk about this in a little more detail, so it allows you to compare 2 or 3 protein samples simultaneously on the same gel. The protein in each samples are covalently tagged with these dyes, and these dyes have no effect on the relative migration. So basically they are very smaller in size, or you can say even if I don't talk about the size, they have no effect in migration. So overall your separation is exactly same like 2D electrophoresis. The only thing is that you can run many sample, like more than two samples, 2 to 3 samples simultaneously in one gel. So generally the proteins that appear on this particular gel are in form of spot, and then you have a fluorescent signal associated with that. And once you have the intensity of your fluorescent signal captured in your image analyzer, then you can analyze your data, and you can study your data in all the 3 samples together. So that is there. And if I talk about the protocol, we'll discuss the protocol as well. But right now it is there. Right now I'm just talking about the different dyes and different things that I used in the DIGE. So next one is amine reactive dye. These are the cyan dyes that are NHS ester derivative, and that they are specifically labeled the eta amino acid of the lysine residue in the protein. So this ensure consistency and stable labeling while minimizing any kind of interference. So are you understanding this thing, that when you have a a dye that is specifically binding to a particular amino acid, that reduced your artifacts, and that reduced your um uh, uh, non-specific labeling. So this labeling is stable, it is consistent because it's binding particular to a single amino acid. So that is there. Then you have minimal and saturation labeling. So DIG typically uses minimal labeling to avoid altering protein properties. So I mean, this labeling will not interfere with the protein properties. It will not change the structure. It will not have any effect on these protein, because the labeling itself is done. Yeah.
Ma'am, suppose there is a protein where we have very less amount of lysine.
Hmm.
So, would the intensity of the dye uh, vary in that matter?
So, hmm.
And another question is, are each of the dyes specific, to lysine only?
No, when they have NHS ester associated with them. So there are some links which are, so you can modify dyes. Okay, so whenever you have a fluorescent, because these are cyanin based dye, that's why they are called CY2, CY3, and CY5. But if you are associating NHS ester, its property become amino in nature. So they basically bind only to the eta amino group, which is only present in the lysine residue. So that's why it is specific. One. Second, you said that if there is a less lysine in your protein, so yes, definitely your signal will reduce. But because this is a fluorescent based, so what will happen? So that that thing going to remain. That when I say that you have a high abundant protein, for say, or you have some proteins which have high lysine content as compared to the protein that has a low lysine content. But in this case, definitely the signal will be less in case of low lysine residue containing protein. um, but here, because it is a particular amino acid based uh, association, you will find spot. The kind of images you are capturing depends upon the kind of microscope or the kind of image analyzer you are using. So if you have a very high sensitive analyzer or high sensitive camera that is capturing your fluorescence, you will even get a very small dot resolved on your gel. That completely depend of the kind of setup you have. But definitely you are right, if the lysine residues are less, the spot will be lighter in terms of fluorescence. It will give less fluorescence as compared to the protein that have high lysine content. That is always going to be that will be there. But because here, I'll talk about this thing in like the complete protocol in the coming slides. But here you are actually doing a combined, if you are going to study a combined effect as well. So when you're studying a combined effect, it will give you more clear picture of your analysis. I mean, you can analyze it in a more accurate manner because you are actually getting the spot. uh, I mean, if the spot is overlapping in that particular point, your last overlap image will actually give you the better resolution of your samples. So I'll talk about it in detail. Right now I'm not commenting on this thing, I mean, exactly here. But when we talk about the workflow of this particular technology, then you'll understand that what exactly I'm saying. So both the questions are quite valid, and they are good questions. So the answer is there. But definitely the proteins, like in 2D, in two-dimensional electrophoresis, when you have an abundant protein and when you are trying to study a low abundant protein, you will always have a mask effect of highly abundant protein. Here also, if the protein is highly have a large number of lysine residue as compared to the protein with a minimum or very less number of lysine residue, the effect you will definitely going to see in your gel. You will get a very um, fainted, I can say, spot in case of the protein with very low abundant lysine residue. I hope I have cleared your
Yes, ma'am. I understand it.
okay, fine. So where were we? Yeah. So also I was talking about the minimal labeling. So you actually use a very less. So these labels are very, as I've already mentioned, they are very specific. When they are associated with some kind of ester, they're specific. You require them in a very less amount. If you have studied about confocal studies or any of the fluorescent dye based study, the amount of dye that you use is very less and it never interfere with the normal processes or normal biomolecules, and it does not hamper your the sample. So when you have a specifically binding protein or protein that is associated with a particular residue, then your resolution is definitely much better, and you will not find more of artifacts in that particular case. And when I talk about the saturation labeling, that can be employed when targeting low abundant protein, allowing for enhanced detection sensitivity. So here you can actually remove the saturated portion, and you will have the protein detected which is low abundant. Because once, if your one of the protein is highly abundant or maybe highly rich in lysine residue, it's detection become saturated at a particular time point. So you have, when you capture any image, you have a time limit on that. When you put that particular time limit and when you go beyond a particular time limit, the proteins that are present in abundance will be saturated. You will never find a difference between their spots. So if for example तीन चार proteins हैं और वो बहुत abundant है और कुछ proteins हैं जो बहुत कम present है, so the protein that are very abundant, eventually when you increase your time for the image capture, these abundant protein will be saturated. You will find no difference in their spot. But when I talk about the proteins that are low abundant, you will have clear cut view of those proteins. You can capture their abundance, you can capture their difference in expression once you have a saturation labeling. We'll talk about it in the coming this thing. So talking about minimal and saturation labeling. So minimal labeling, one of the small fraction, usually less than 1% of the available lysine residues are labeled with the fluorescent dye, ensuring that protein biochemical properties remain largely unchanged. So why, as the name suggests, it's minimum. So you use such a minimum diet that mostly very small fraction of the lysine residues are labeled. So imagine if you are labeling your sample with a very small amount of any dye. So it will bind to lysine residue, and but it will not, it will, there will be some residues which are not bound to these dyes. So minimum labeling, so usually it's less than 1%. So you have better picture because jitna kam wo because it's a specific labeling. So definitely you're going to bind to lysine residue, probability is very less that it will leave any protein in that particular sample. It's very less. So imagine you are labeling it in a very low concentration. So when you run your gel and when you're going to capture the picture uh, in your image analyzer, uh, at that particular time, you will have a very clean and resolved spots for these proteins, right? So minimum labeling is that. Then you are maintaining proper integrity. So minimum or saturation labeling क्या कर रहा है? By labeling a minimum number of lysine residue, minimum labeling preserve protein function and mobility. Because now it's not. So एक बार अगर आप imagine करोगे protein structures को, so if your dyes are bound in the in the protein chain, because protein contains of amino acids, it's a polymer of amino acids. So if some dyes are bind to a particular amino acid, so what will happen? It will change its mobility. It may change its mobility if all of the lysine residues are bound to these dye. So imagine how heavy it will become if all the residues are bound, because lysine is a very commonly present residue in the proteins, right? So अगर सब पे बाइंड कर जाएगा तो उसकी मोबिलिटी पे डेफिनेटली इफेक्ट पड़ेगा. सो व्हेन यू डू मिनिमम एंड such kind of labeling, minimal and saturation labeling, then you are actually reducing that chances. You're not letting your protein getting affected by these bound labels. सो मोबिलिटी इफेक्ट नहीं हो रही है. अगर मोबिलिटी इफेक्ट नहीं हो रही है एज वेल एज बाइंडिंग बहुत लिमिटेड है, सो उससे क्या हो रहा है, योर आर्टिफैक्ट्स चांस जो है वो रिड्यूस हो रहा है. वाई? बिकॉज इट इज बाइंडिंग टू अ वेरी स्पेसिफिक रीजन ऑफ दैट पर्टिकुलर प्रोटीन, and that is maintaining the separation accuracy. How? Because when the binding is less, आपकी resolution, everything going to be very high, and then reproducibility also become high. So that is there. Then saturation labeling purposes, basically as I have told you, जो abundant protein होते हैं वो सारे saturate हो जाते हैं. Saturation labeling target available lysine protein in residue in proteins, providing higher labeling efficiency and sensitivity, especially useful for detecting low abundance in the complex sample. Then we come into increased detection sensitivity. Definitely when you're using the saturation labeling, you are basically trying to get low abundant proteins to be visualized when you are detecting your sample. right? So making it possible to visualize those particular protein. And when you are doing a minimal, because saturation में तुम ज़्यादा amount use करो, you actually saturating the high abundant proteins, or proteins that are getting that are present in a higher amount as compared to other proteins. But this may not happen when we are talking about minimal labeling. But in case of minimal labeling, you will have resolution of maximum protein there. Low abundant protein might not be labeled, because when you're using a very less amount of dye, that is usually when you're labeling only 1% of the lysine residue, imagine the protein that is present in a very low amount and the protein that is present in high amount. So high amount में चाहे लाइसिन का लाइसिन रेजिड्यू कम ज़्यादा भी present है, but because you're doing minimum labeling, so only 1% will have the label. But if I talk about the low abundant protein, which is already present in the sample in a very low amount, its lysine residue, additive effect of its lysine residue will be less as compared to high abundant protein. यू गेटिंग माई पॉइंट कि एक मिनिमम लेवल पे कर रहे हैं, एक सैचुरेशन लेवल पे कर रहे हैं. सो सैचुरेशन लेवल पे क्या हो रहा है, जो बहुत ज्यादा अमाउंट पे प्रोटीन है वो सब तो सैचुरेट हो गए. नाउ देयर इज नो डिफरेंस इन देयर intensity of these spots, but the proteins that are present in a low abundant abundance, they will be clearly spotted on that particular gel. So these are different kind of strategies when you are studying a different aspect of the protein. So if you want to understand, if you want to looking for biomarkers, you will go for saturation level. And generally biomarkers are produced by, I mean, when the discovery of biomarkers generally happen with low abundant protein. So if I talk about cancer, if I talk about any other multi-gene disorders, the chances of getting a good biomarker is with associated with low abundant proteins only. उसी से पता चलता है, क्योंकि वो इतने स्लाइट चेंजेस हुए होते हैं और वो ऑलरेडी इतने कम प्रेजेंट होते हैं, तो उनको डिटेक्ट कर पाना कभी कभी बहुत डिफिकल्ट भी होता है. बट एट द सेम टाइम दे गिव अ वेरी एक्यूरेट रिजल्ट्स इन व्हेन वी टॉक अबाउट डायग्नोस्टिक्स. Fine. Then there's a trade-off. This is one of the thing that is done during DIGE. While saturation labeling offer great sensitivity, it may offer alter protein migration. Why? क्योंकि saturation में we are using high amount of labeling. सैचुरेशन लेवल पे लेबल कर रहे हैं, तो अब यहाँ पे क्या होगा, माइग्रेशन लो हो जाएगा, बिकॉज बहुत सारे लाइसिन रेजिड्यूज आर नाउ एसोसिएटेड विद दीज डाई. वेयर एज व्हेन वी टॉक अबाउट मिनिमम, उसके केस में क्या हो रहा है, उसके केस में यू आर नॉट गेटिंग गुड labeling density. Sorry, you are not getting good differences between the low and high abundant protein. So here there is a trade off, because वहां पर अगर हमें लो अबेंडेंट प्रोटीन की डिटेल्स मिल रही हैं, बट इन केस ऑफ मिनिमम वी आर नॉट गेटिंग दीज डिटेल्स. सो दिस इज काइंड ऑफ अ ट्रेड ऑफ, क्योंकि एक किसी चीज में बेटर है और एक किसी चीज में बेटर है. सो दैट्स व्हाई आई हैव ऑलरेडी मेंशन, and I have been telling you this thing from the day one, that in case of proteomics, depending upon what kind of a result you want to get or what kind of a study you want to do, you choose or you make your own workflow. So here also, if you're looking for low abundant protein, you will go for saturation labeling. So these are some terms, and talking about how it is done. So if you could see here, in case of minimum labeling, you use, because it will bind to lysine. In case of saturation, it will bind to cytosine. So sorry, cysteine. Depending upon the kind of dye you are using, you may change these properties. But here, the time as well as the kind of dye you are using and the time you are giving for the labeling is different. So दोनों में तुम्हारा प्रोटीन डाई वही बन रहा है, like you are getting a complex of protein, I mean dye associated with your protein. But when you're going to run in case of minimum, you will have uh, less intensive spots but more in number. But when I talk about saturation labeling, you will get more saturated spot with less uh, I mean, the saturated spot, the intensity of spots going to be very high in some cases, and you will have a better picture of your low abundant protein. So this is there. They use different. I mean, this is not telling you much, but basically telling you that kind of protein, the amount of protein that you use per dye. So if you could see here, in case of minimum, you are using a very less amount of dye, that is 400 picomolar. Picomolar is, can anyone tell me what is the dimension of picomolar? I mean, is it 10 raised to power what?
Uh, 10 power minus 12.
Yeah. And what about femto? Sometimes you use dye at femtomole level as well.
10 power 14, ma'am.
15.
Oh,
So it's can you imagine that for such 15 UG protein, which is a huge amount of protein, you're just using 400 picomole, and sometimes it goes as less as femto, depending upon the kind of work you are doing. So that is there in case of minimum, but in case of saturation labeling, you are using excess of dye, so that the cysteine, all the cysteine residues are labeled. So the proteins that are highly abundant will have much higher intensive spots. You are not bothered about those protein. You are trying to capture the low abundant protein. So you will have a clear spot resolution for low abundant protein, while your high abundant protein might have some saturated spot or merged spot. Uh, so that's how you use these two labeling depending upon your need. Now, talking about labeling chemistry, that how these dye bind to each other, what kind of a bond they have with what particular, like I have mentioned that if you're using lysine based binding, where you have your NHS group associated ester group, which actually bind to a lysine residue, eta amino group of the lysine residue. So this is basically talking, this is what we say lab, is labeling chemistry. So what kind of a chemical bond they are making with the protein is what we're going to study here. So cyanine dye core structure has cyanine dye backbone, and it consists of polymethylene chains connecting two nitrogen containing heterocycles, which provide tunable fluorescence property across the different wavelengths. So for CY3 it's green, for CY5 it's red, and you have a combined effect as well. Now talking about reactive NHS ester group, which I have already mentioned in the previous slide, that it has a specificity with the eta amino group of your lysine residue. So this is basically an addition to the cyanine dyes. So if you are using NHS ester associated cyanine dye, it will bind to lysine. If you are using different different association that will bind to a different type of amino acid, you may make some variation depending upon the availability of dye. So conjugate कुछ भी करवा सकते हो इसमें, so that आपकी binding किसी particular amino acid से हो, like in case of NHS, it's binding to lysine. Like, so this is there. So these are the dyes, actually different modification in your cyanine dye, which is giving you a particular kind of binding. Then amide bond binding, NHS ester reacts specifically with the eta amino group of the lysine residue in protein under mildly basic condition. So अब ये bond जो formation हो रहा है, ये basic condition में हो रहा है, forming a stable amide bond that securely link the fluorescein dye to the protein. So now you getting, when you're using NHS ester group, your buffer needs to be basic in nature. So these are the things that you have to keep in mind. That's why I have been mentioning this for a very long time, that you have to design your workflow depending upon what you are using and what kind of result you want. So proteomics is very flexible in terms of making your workflows. You may use one kind of a technique, so you can start with the gel based approach, And you may directly come to techniques that are associated with gel-free approaches. So you can mix match these techniques and make your own workflow depending upon your requirement and availability of the samples. Then you have minimum structure interference, as I've already mentioned, because it's binding to a particular residue, as well as you are using a minimal labeling, it will not affect the overall mobility, charge, mass of the protein. So basically it's preserving its native electrophoretic properties and ensuring there is an accurate separation during gel electrophoresis. Then you have optimized spectral properties. So polymethyl methine chain length and substituents in the hydrocyclic ring of these cyanide dyes determine their excitation and emission. So इनकी जो spectral property है, that is also very optimized and very well established, allowing for multiplexing with the minimum spectrum overlap critical for accurate DIG analysis. So because they have a very specific wavelengths, all are already they're already determined, they're already established. So when you're doing multiplex studying is when you're studying three different samples together. In that is the case of DIGE. You're using three different fluorescent dyes in three different sample, and you are studying them together. Then multiplexing images due to their specific excitation and emission are more accurate, and they give you a clear cut picture, and it's easy to analyze when you're performing multiplex DIGE because of these properties of dye. So talking about the chemistry of this is basically same. So you have living cell, you have your control cells. Okay, so you are just do the cell lysis, and you will allow your proteins to come out. You'll make the lysis, protein lysis, and then you make them react with a particular protein. So for here, you're using CY3 CY3 dye. And here you is using CY5 dye. So you have one of the control sample, one of the test sample, and when you do the 2D, you will mix them together. They both, so the test sample protein are labeled with red dye, and your control sample protein are labeled with green dye. So when you're going to run your sample, you will get three different spots. One is red, another one is green, and there is yellow. So yellow are the spots that are present in both the samples. These are the protein that are present in both the samples. Red will give you protein that are present in your test sample, and green will give you the protein that are present in your control sample. So now you can properly, you can easily detect what are the proteins that are present specifically in your test sample. You can excise the protein, you can digest these protein, and you can send them for mass spectrometry to understand what all proteins are present in test only, and what all protein that are present in your control only. Combined ka, because they are present in both, so you that might not be of your interest, but sometimes it is also of the interest, because when you're looking for the common proteins, then you may excise that particular gel region as well. So this is basically what happened in DIGE. This is again the same kind of a cartoon where you're using a proteome A, which is a one set of protein sample, and then you have a proteome B, which is another set of protein sample, and you're labeling them with a different color and running on the same gel. So that is definitely the same thing. Then coming to the workflow, or you can say the experimental strategies that we are using during DIGE. So here, if you could see, the first lysate is labeled with CY2, that is yellow in color. The another lysate is labeled with CY3, and then the third lysate is labeled with the blue color. So red, yellow, blue. Then you are mixing these labeled lysate and running on a single gel, and you get three different images from there. सोचो कि एक ही जेल चलाया, कितना टाइम बच गया, कि 3 टाइप के सैंपल्स तुमने एक ही जेल में रन कर लिए, एंड डिपेंडिंग अपॉन डिफरेंट एक्साइटेशन एंड एमिशन वेवलेंथ you will capture 3 different images from the same gel. And then you can do overlapping, then you can do image analysis. Overlapping images kar sakte ho, in gel analysis differences dekh sakte ho, because you can definitely have the normal, after taking the fluorescent, you can definitely stain these cells and can have a proper Kumasi stain as well. And you can definitely do 3D analysis, where you can study the additive as well as single protein effect. So sometimes you will have an overlap. So in overlap, you will have an additive effect. But if you have a specific protein specific to a particular lysate which is not present in other lysate, then you can excise that protein and can study further that what all changes are there in these samples. So this is done. Then you have a proper clusteral. When you once you have your sequences or your mass spectrometry done, you do a cluster analysis and you study what all proteins are present in your sample. And you may also do 2D Western blot uh, after picking a particular spot or a particular region to resolve your gel more. So this is basic workflow that is very uh, simple to see, but is complicated. But at the same time, it is giving you a complicated result. You're getting 3 sample result in one gel, which is not possible when you're doing only 2D gel electrophoresis. Because here you're using fluorescent dye, and that is what you're exploiting the properties of fluorescence here. So this is again the workflow, and if you could see here that sometimes you can also use an additive pool. Okay, so this is different type of a workflow where you're using a pool internal standard use कर रहे हैं हम, जिसमें A plus B का pooled common region है. Then you have protein extract A, then protein extract B. इन सबको लेबल को मिक्स किया, 2D जेल रन किया, CY2 मिला, CY3 मिला, CY5 मिला, 3 images capture of the same gel with the different different fluorescence give you exact effect of different your extracts. And then you can overlay and you can get to know that what all proteins are present in each of the sample, specific to each sample as well as common to these samples. So this is just the same thing. I'm just moving ahead because I think you guys have seen seen the videos. uh, Sir has taken the help of a person who is actually helping you to understand the analysis of this particular technique. But I'll try to give you an overview here. You may have the detail of these analysis in those videos only. I just have 10 to 15 minutes left, then we have to move to the question and answer. But still, I'll try to give you a small brief कि कैसे analysis करते हैं इसमें. So for example, you have 3 different labeling करने के लिए आपके 3 different dyes हैं. So this is one of the example here. They have taken actually how do you do the fluorescence difference in gel electrophoresis. So they have used approach where they are using 2 dyes or 3 dye difference in so 2 dye DIG कर रहे हैं इस केस में, और यहाँ पे 3 डाई यूज कर रहे हैं. जिस केस में 2 कर रहे हैं उसमें क्या कर रहे हैं, दे आर यूजिंग नॉर्मल मसल प्रोटियोम and then pooled internal standard with the different dye, and then you have dystrophic muscle proteome and then pooled internal sample. So they're just using 2 dyes. One is normal, one is dystrophic. Okay, and they are then mixing these sample. This will run on a different gel, this will run on different gel, and then once they are run, they will capture the image and they try to overlay on each other to know what all proteins are present in the normal one and what all present proteins that are present in your dystrophic muscle proteome. So this is 2D, 2 dye based DIGE. Then you have 3 dye based DIE where you're using normal muscle and the pooled internal standard is taken as the third, with the third is associated with the third kind of a dye. So यहाँ पर एक ही जेल रन करना पड़ेगा तीनों को मिक्स करेंगे और you will get the image. इस वाले केस में you have to run two gels. So this is here. Then multiplexing is basically when you are reading, you are doing different samples on a single gel. So you are using 3 dye, or sometime nowadays people have started using 4 dyes as well. So in 4 dyes के अंदर different colors के basis पे 4 dye होती हैं, 3 dye होती हैं, and then you give each sample each dye, and then you run it together, and then you do a multi channel scanning of these of this particular gel. So here image acquisition and alignment कैसे होती है? After 2D gel electrophoresis, fluorescent image of the gels are captured by each dye. So you will have 3 different images. One is red, another one is yellow, and then you have a blue. So you will have 3 different images that may have different pattern. But when you're going to overlay it, you will get a proper image, and then you can actually understand the differences between the three samples. So images are then aligned to the correct for any gel distortion and ensure accuracy spot matching across the samples. So it is done. This is how they acquire this particular image. Then you have spot detection and quantification. So software automatically detect protein spot on the gel and quantify their fluorescence intensity. So अभी तक हमें सिर्फ डिटेक्शन हो रहा था. Now you're quantifying as well. तो quantify कैसे करते हैं? Depending upon the fluorescence intensity. If the intensity is high, the protein is present in higher amount. If the intensity is low, the protein is present in the lower amount. So the intensity of each spot reflect the relative abundance of the protein in the each label sample. So for example, same spot पे red भी आया है, same spot पे blue भी आया है, same spot पे green भी आया है. That shows that this particular protein is present in all the 3 samples, and depending upon the kind of intensity, even if it's an additive color, because ये combined color होगा, but combined color की भी intensity कितनी है, उससे आपको पता चल जाएगा कि red वाले sample में कितना प्रोटीन है, blue वाले sample में कितना प्रोटीन है, और yellow वाले sample में कितना प्रोटीन है. So these kind of quantification you can easily do using DIG multiplex during DIG multiplex analysis. So normalization using internal standard. Internal standards generally आप label कर देते हो C/2 से, and is used to normalize spot intensity across the different gel. Just give me a minute. I'm having a call. I have to take this call. Just give me 1 minute. Thank you. Thank you for staying with me. So yeah, so normalization is done using internal standard. So here internal standard, basically it has an pooled effect. It give you the average of all the samples. So it will help you understand. So you generally normalized your intensity with the help of internal standard, or internal standards are basically pooled effect of these samples. सो क्या किया, एक को रेड दे दिया, एक को ब्लू दे दिया, एंड दोनों के मिक्सचर को ग्रीन दे दिया, राइट। सो नाउ जो दोनों के मिक्सचर को ग्रीन है उसकी इंटेंसिटी से हम नॉर्मलाइज कर रहे हैं फॉर रेड को भी और ब्लू को भी। सो इट्स डन सो दैट यू कैन इजी, एक तो आपको स्पॉट की इंटेंसिटी एंड स्पॉट की प्रेजेंट एक्यूरेटली पता चलती है, as well as you can normalize using these standards. So it minimizes the technical variability and allowing for a more accurate comparison of protein expression level between the samples. So that is done using internal standards only. Then statistical analysis for differential expression, you use a tool to compare the protein expression across the sample, identify the spot with a significant difference, and the common approach is generally using statistical tools like ANOVA, t-test, fold chain analysis to detect the differentially expressed proteins. Then you have protein identification via mass spectroscopy. So for example, I have already told you that if you are looking for a biomarkers, so if you have a normal versus cancer sample and you get to know that these are the protein that are present only in your cancer sample, then you can excise that particular region of the gel, digest it, analyze it in mass spectroscopy. You'll get the sequence and you can identify that these are the protein that are only present in cancer sample and are not present in normal sample. So by that you can actually get a better biomarkers for your study. Then what are the advantage of DIGE? So it has a high reproducibility because you're using internal standard here. It is very consistent, high reproducibility as running all the sample under identical condition on a same gel. अब क्या हो रहा है यहाँ पे reproducibility high होगी because now you are running the samples on the same gel. So if you have 3 samples to compare or if you have 2 samples to compare, you can run it in the same condition on same gel and get result for both. So it actually increase the reproducibility and improve the results consistency. Then it has a dynamic range. How? Because you are using multiple sample together. So you can use up to 3 samples and you can label them with a different dye, analyze them together on a single gel, enabling direct and effective comparison. Then you have accurate quantification with the internal standard. How? Because you're using the internal standard. Internal standards kya hai? They are the pooled of both the samples. They are the, I mean, you're using same amount of A and same amount of B, pooling it together and giving it a color, giving it a different color. So additive जो है आपका, that is internal standard. And इससे when you are comparing, you have very good accurate quantification in that case. So here variability will be minimized and it will enhance your precision in quantitative comparison. Then coming to enhanced sensitivity for low abundant protein. So saturation labeling increasing the detection of low abundant protein, offering better insight into the complex samples. So up saturate करके जब कर रहे हो, so definitely you will have a better resolution of the proteins that are present in low abundance. Then you have reduced gel to gel variability because why? Because you are using one gel across 3 different samples. एक ही sample को एक ही 3 samples को एक ही gel पे run कर रहे हो, you are actually reducing the gel to gel variability. Then it is robust quantitative analysis because fluorescent dye provide a wide dynamic range and stable signal. Why? Because they are specifically binding to a particular residue, giving you a better quantification of your proteins. Then definitely there is a limitation. It's a very costly as compared to a normal 2D gel electrophoresis. Why? Because you're using fluorescent dye. Fluorescent dye are generally costly and that makes this particular technique very expensive. Then you require specialized equipment because you have to have all the filters. You have to have an imaging system that will give you high resolution. So your laser should be there, your high resolution imaging system must be there. So you require high or high cost or specialized equipment for these to capture the images for these gels. Then you have a complex setup because you have to make your samples should be made in a particular manner. Handling should be done in a very precise manner, which make it more technically challenging. So this particular technique is protocol intensive, and you have a lot of things that should be done in a particular manner. It has to be made in a particular way. So that is making it more complex as well as difficult to handle and technically challenging. Then you have sample limitations. So you cannot go beyond 3. People go for 4, but that is all basically a pooled effect. Okay, so you cannot go beyond 3. So ये एक issue है. Sometimes your spectra overlap. So क्या होता है कि when you have additive effect, when you are doing an overlay study, or you have a pooled effect, तब क्या होता है, तब red, blue और yellow, because you have to capture all the thing, तो spectra आपका overlap करता है डाई में, and जिसकी वजह से quantification accurately नहीं हो पाती. So these are some limitations, but at the same time it is giving you a very good resolution in terms of looking for biomarkers, running 3 samples together, and very low gel to gel variability, thus making it a reproducible technique. So that's all about DIGE. Now I'm moving to system biology. If you have any question regarding DIGE, please turn on mic and ask. Just give me a second. I'll be back. But meanwhile, if you have any question, please ask. I'm assuming you don't have any question. Coming to system biology, how many of you are aware of what exactly system biology is? I want answers. Please make it a little more interactive. It will be fun. I mean, proteomics is a very good subject and you are learning a new thing here. So please make it more interactive, then only you'll have a better picture of this.
Ma'am, my understanding is like in system biology, we focus on like quantitating biological data, like making it more data science oriented.
Okay, okay. Oh, my mic was turned off. Sorry. I just realized because you were not answering. Okay, fine. So, system biology, basically, you were somewhere right. There are a lot more things to it. But when I talk about system biology, basically, you are understanding biology or biomedical processes uh, uh, to understanding the larger picture. Basically, it's a biomedical approach to understand the larger picture, be it an organism level, tissue level, cell level, by putting its pieces together. So you are actually understanding the whole system, where your smallest part, which is cell, and organism, how these levels are linked, how this whole, because what is happening in an organism, things are working, you know, uh, cell is working, these cells are working together, so your tissue is working, then organ is working, then whole, all these organ working to make an organism to work, right? So if you see things in that way, you will know what exactly system biology is. So it is it, and with system biology, you are trying to actually understand with the help of computers, with the help of mathematics, the whole complex biological system, right? So I mean, if you see life or if you see the kind of biological system happening in an organism, be it plant, be it animal, you start with the basic unit, which is cell, right? So cell is doing some work, and one cell is doing some work and making it I mean, active or live because it's working, right? And then all such kind of cells working together, saying that the tissue is working, then organ, then organism, right? So the system is quite complex. And when you try to try to understand this system in a computer, in a computational modeling manner or a mathematical model से जब आप पढ़ने की कोशिश करते हो, that is system biology. So you can say it takes a holistic approach to understand how organisms interact at a different scale, including microbes, including plants, including ecosystem, including animal. तो उस वे में सोचो, सबसे टाइनीएस्ट चीज से लेकर सबसे bigger picture tak, when you try to understand and combine them together with engineering, with physics, with computer science, with mathematics, and try to simulate that in a system, try to understand the system with the help of these area of science, is what system biology is. Okay, so it is basically a collaborative work of different disciplines, where you are studying cell biology, where you are studying genomics, proteomics, transcriptomics, immunology combined, and to understand what exactly is happening in a system manner. It's a vast thing to talk about. So right now, अगर इसका एक overview दूँ तो it's like this: the biology combined with technology and computation is system biology. Is all the three things together give you a picture of how the system is working. And system can be a cell, a system can be a tissue, system can be an organ, organism, ecosystem, different organism interacting with each other. You can define system in any manner. Okay, so you are studying basically so many things together when we talk about system biology, with the help of physics, engineering, mathematics. You're studying biology. So that is the beauty of this particular technique, or you can say field. So when we study system biology, what are the proteomics technology that we utilize? So we utilize different antibody-based technology, studying in when we are, as I've already mentioned, it can be a cell biology, it can be a proteomic study, it can be a transcriptomic study, it can be immunology study, right? So you use some proteomic technology when you are studying a system biology, because here you are actually using computers as well as mathematical models to understand the system. So sometime you applied some proteomic technology to understand the system biology, and such technologies are ELISA, because they are antibody based, generally antibody based. Antibody based is basically to understand what are the antigens or what kind of immunological responses are happening at that particular level. So you use immunoassay based multiproteomics technique. It may be microarray, it can be Western blot, it can be ELISA. So generally what happen, you do a array based studies. An array में generally क्या होता है, array is a system. It's like a system here. So in one particular array membrane, you have a particular antibody that is capturing your target analyte. And once it is captured, it is then bound to some substrate, some fluorescent dye base. I mean, just anything that can detect this particular interaction, where you have a capture antibody and you are targeting a particular analyte. So these are the techniques that you use. And once you have your captured analyte or a pool of captured analyte, you take out this particular pool or this particular analyte and study this using proteomics. So this is there. So microarray is one of such techniques that we utilize in system biology, and it's a proteomic based study. So initially microarray, when it when people or researchers started doing it, it was DNA microarray, and it lies on the principle of hybridization. So the principle of DNA microarray is basically hybridization between the nucleic acid strand. So one of the strand is bound to your array, like in this case you have something bound. So it is, something is bound, either it's an antibody, either it's a DNA oligos, something is bound which is binding to one analyte present in your sample. And once it is bound, you're getting a signal, and that signal ensure that the analyte you are looking for is present in your sample. So likewise here, in case of DNA microarray, you're using the property that is used is basically hybridization between the nucleic acid strand, and the property of a complementary nucleic acid sequence is to pair specifically with each other. So you have a particular specific oligo bound to your array, and you are detecting a particular DNA fragment in your sample. So you have, so if you could see this cartoon, you have a healthy cell and you have a pathological cell, some disease cell, and you're taking out the DNA, and you are taking out the DNA or RNA, isolating it. You're taking out the RNA, and then you are reverse transcribe this particular RNA and making it a cDNA. And once your cDNA is made, you are fluorescently tagging it, healthy with green, diseased with red, and then you're going to run it on your microarray. Fine. So different analytes, different oligos are present in on each array. If your that particular analyte is present on both the in the both the sample, you'll have yellow color, which is basically present in both cells. And if you have red or green signal, it will give you the sample that is only present in healthy or in your diseased. So this system is basically DNA microarray, and that's how you detect what all sequences are altered or changed or present in healthy as well as pathological sample. So at least two samples are hybridized to chip. Complementary nucleic acid sequence between the sample and the probe attached to the chip get paired via hydrogen bond. The nucleic non-specific binding sequence while remain unattached and washed out during the washing step. So once bound one are there, but when you do the washing, the one which are not bound with the oligo that is present on your array are washed off during washing process. Then the fluorescently labeled target sequence that bind to a probe sequence generate a signal, and the signal depend upon the hybridization condition. You have to maintain a buffer as well as temperature condition so that your binding happens. And then washing after hybridization, etc., while the total strength of the signal depend upon the amount of target sample present. So अगर signal बहुत high है, in case of red, green or yellow, if the signal is high, that means the amount is also present in a high, like the amount of a target sample is high in your target protein is, or target DNA sequence is high in your sample. So using this technology, the presence of one genomic or cDNA sequence in 1 lakh or more sequence can be screened in a single hybridization. So एक ही पूरे उसमें आप इतनी सारी sequences को screen out कर सकते हो. So this is the beauty of DNA microarray. Likewise, you have a protein microarray. So for protein microarray, protein arrays, or you can say they are also called protein chip. It is a process of immobilizing numerous protein on a solid support using miniature assay that describe protein interaction. So it can be antibody screen, it can be enzymatic screen, it can be protein DNA interaction, it can be protein interacting with a small molecule, protein lipid interaction, and protein protein interaction. Depending upon what kind of a interaction you want to analyze or study, you use different base or different array molecule which are bound to your solid support. So it's a powerful methodology in a high throughput format to analyze thousands of the known protein in a single experiment. You can analyze 1,000 proteins in a single experiment. An indispensable tool for proteomic application, multi-parameter clinical diagnostic test can be done in one go, and many other fields. So these are just application. So protein microarray are of three types. One is analytical protein microarray, which is basically analyzing. They are mostly representing by antibody microarray and can be used to profile a complex mixture of protein in order to measure binding affinity, specificity and protein expression level. So analytical में हम analyze कर रहे हैं. So we are using a library of antibodies or aptamers uh, on a glass slide, and the targeted protein will be detected either by direct labeling or a reporter antibody in a sandwich assay format. So generally ELISAs and other things are done in that way. Like basically you're using either a direct fluorescent labeling here, or you are using some uh, substrate is associated with your detection antibody, and then uh, some enzyme is associated, and then you are giving a substrate. So it's a sandwich assay format. Then you have a functional protein microarray. They are constructed using individual purified protein that involve full length functional protein or protein domains. So these are done basically to understand the full functional protein or protein domain present in your sample. This class of array enable the study of biochemical properties of the protein, such as binding affinity. बाइंडिंग एफिनिटी बताता है, प्रोटीन प्रोटीन इंटरैक्शंस बताता है, बिकॉज यहाँ पे हम फंक्शनल एस्पेक्ट देख रहे हैं प्रोटीन्स का, एज इट इज कंप्लीट प्रोटीन्स आर बीइंग यूज्ड. Yeah, so you are actually detecting binding activity like protein DNA binding, protein protein binding, protein peptide binding, protein drug binding, or protein lipid binding. So here you are analyzing the functional aspect of the protein. Then you have reverse phase protein microarray, which are called RPAs. They employ an opposite array format by immobilizing many different lysate samples such as tissue, cell lysate, fraction cell lysate on a chip. So you are actually immobilizing the complete lysate on a chip, or complete tissue on a chip, or a fractionated lysate on a particular chip. And many different probes can be tested to specifically identify a specific protein in these lysate samples. So RPAs are frequently detected with the fluorescent chemiluminescence or calorimetric assay, allowing for determination of post-translation modification to define alter protein as a result in the disease. So this is basically when you are analyzing whole lysate against a particular protein. So that is reverse phase protein microarray. And here you use chemiluminescent or chlorine, because a slight change will tell you that that particular protein is either altered, or it has I mean, if it's not giving you the color, that means its enzymatic activity is either altered, or if it's giving color, that means it is getting activated. So such kind of modification happening at a protein level can be detected using reverse phase protein microarray. Then biomarker identification using protein microarray, as I've already told you कि एक particular sample है which is healthy in nature, and then you have a tumor or a disease sample. You are putting it on a array on your mic. You are giving a different label to these sample. One is green in color, one is red in color, and when you are running your microarray, and then you are getting samples that are, then you're getting a spot or a genes against a particular protein that are only present in your normal sample. Then there are some protein that are only present in your disease sample, and then there are some common proteins. So from the common proteins, you will get to know that these proteins are present in both the condition. But from the diseased and the one that are present in the normal, you can get a good biomarker. So you have to identify depending upon your particular game. I mean, you will get lot many spot, but then you have to validate your data to identify a particular biomarker with different samples. So that's all from my side. Uh, this was a brief of system biology. Definitely we're going to study System Biology and Proteomics in coming classes. This is just a brief for you guys. So far, any question? Shall I move to the previous year week 4 assignment actually? Sorry. So this is week 4 assignment. Uh, yeah, so it is previous year week 4 assignment. So this is the first question. Oh, I'm so sorry. This is actually from here. I may have an extra slide here. Yeah, so let's start. So the first question is, shall I start with the questionnaire, or if you have any question, please turn on your mic and ask. Okay. So the first question is, Kush conducted a 2D DIGE, that is differential in-gel electrophoresis experiment, to compare the proteome of the control and the tumor sample. He used Psi3 dye to identify the control sample and Psi5 dye to identify the tumor sample. Below is the representation of image analysis result combined with the 3D depiction of a protein A. So this is basically a 3D depiction of protein A here. So this one is normal and this one is tumor, right? So the green one is C by 3 actually. So this one is control and this one is your tumor. C by 3 is green, right? C by 5 is red. So the expression for protein A is unchanged. Protein A is upregulated to tumor sample and protein A is downregulated in tumor sample, or protein A is differentially expressed in control and tumor sample. So what conclusion may be drawn from this study?
Ma'am, I have a question regarding the dyes here. Um, is C3 red?
Tell me, मैंने अभी
I think
आपको
I think C3 is red and C5 is blue, but here C5 is green. I'm not sure.
C3 is green if I'm not wrong.
Mm, okay.
So C by 3 is green. 3 is green in color. The C by 5 going to be red then. So if your control sample has, this is your control sample. Basically, this was the catch of this question. You must be aware of that C by 3 is of which color and C by 5 is of which color. So this was the catch. If you have the understanding of these dye, then you can do this question. And I've already told you that C by 3 is green in color. So it's generally uh, orange in color as a dye, but it's fluorescent at a green wavelength. Okay, C by 3. Likewise for C by 5, it generally fluorescent at red region.
Ma'am, I remember in the slide that you showed, I think you had mentioned CY3 is red and CY5 is blue.
The color of dye is red,
The excitation
but
is
the excitation is what you have to.
okay.
So that is the catch in this question. You have to remember the excitation. So CY3, it's generally orangish in color or yellowish orangish, but it gives you green fluorescent. Likewise, CY5 is, I think, bluish in color, and then it gives you red fluorescence. I'm looking for answers. What conclusion you can make from this particular image? Itishri,
Ma'am,
you have an answer for this? Yeah, Hari, you go ahead.
ma'am, I think it's C.
C.
Yeah.
So CY3 is control, right? This is control, this is tumor, this is down, this is up. So protein is upregulated in your tumor sample, right? So I don't think so. It's C3 is wrong because it is not down regulated in your tumor sample. This is control. CY3 is control. And CY3 is green in color. This is control,
Okay.
this is tumor.
Okay.
Then statement
Okay.
2 is right.
Yeah, ma'am. I get it now. It should be B then. I
Then
was a
B,
little
yeah,
confused
because
between
P
which
protein
the.
A is differentially expressed in differentially, that is there is a difference between control and tumor, yes. And what is the difference? That it is upregulated in your tumor sample. The answer here is B. Then next question. So there are two questions associated with this particular picture. It says a collection of sample from Sanjoy has two distinct condition marked time point A, time point B. So दो condition है which is marked as a time point A and time point B respectively. He intended to use a DIG experiment with his sample, labeling those from time point A with psi 5 and those with time point B with psi 3. The internal standard employed with psi 2 SDS page were collected for both pools of individual label sample and individual samples. The gel was then examined to identify the protein meant afterward. The combined image of the gel that was produced after scanning at the necessary wavelength is depicted below. Which of the following describe how these gel lanes 1 to 5 should be matched with the various type of run sample? So आपको ये बताना है अभी. I think this is also based on your uh, excitation because this is the final image that you are getting from the analyzer. So these are fluorescent. So can anyone, I mean, you take your time and try to solve this thing. So first lane going to be red. First is red in color. Second is green. Third is white. Fourth is blue. And fifth is yellow. Answer quickly. We don't have time. We have to solve all the questions.
Uh, ma'am, is it D?
How?
Um, obviously 1 and 2 is C5 and C3.
Yeah.
The yellow one should represent the pooled sample, 3 and 5.
Yeah.
Um, I think it should be D, ma'am.
I think you use the smart approach here. Yes, the answer going to be the, oh, it's already labeled. Oh, okay. Anyway, yes, the C, as we have already discussed, that CY5 give you red color. So first sample going to be CY5. Then you have CY3, that is green in color when it fluorescent. So fluorescence of CY3 is green. The second is green. Then their additive is yellow. So fifth going to be yellow, yes. And the additive of all the three dye, red, green, blue, is white. So third one is the additive of all the three pool. Then you have CY2, that is blue in color. So CY2 give fluorescence at blue wavelength. So that is how the answer is B. Clear? Any doubt? Okay, next question. Which of the following does the given images yellow and white band represent? Protein that are present in both samples from time point A and B are represented by the two color band. Protein that are present in the time point A sample data are represented by both color band. Only the protein present in the sample of time point B are present by the yellow band, while the protein present in the sample of time point A are represented by white band. Only the protein present in the sample of time point A are represented by yellow band, whereas the protein present in sample of time point B are represented by white band. So which answer is right here? So basically, they're talking about the yellow and white band, which are additive effect of these dye. Okay, so they are the pooled sample. So answer, I have already given you the
It,
answer.
it should be A.
Yeah, it has both the samples of time point A and B represented by the two color bands. Then coming to this question, for the tumor sample, Palak used CY5 while for the control sample she used CY3. Actually, I have already so this the answers here going to be on the basis of the excitation or the fluorescence they are giving. उसी से सारे आंसर आ जाएंगे. She also created an internal standard with in which CY2 label was applied to the pool sample. So यहाँ पे CY2 जो है वो pooled sample हो गया हमारे control as well as tumor का, right? Then further it is given that merging sample with Psi2, 3 and 5 label also made a pool. अगर आप इन तीनों को भी मूल कर दोगे इसका भी एक pool बनाया उसने. On the SDS gel, the three samples that have been individually and collectively tagged with CY2, CY3, CY5 were run. Only these three samples were run. The gel was scanned after it had run at three different wavelengths, 473, 532 and 635 nanometer, and the gel image was given below. So this is the gel image. उन्होंने pooled डाला, उन्होंने control डाला और उन्होंने tumor डाला. Now, which of the following lane contain only the control sample? Control, only control किस में दिख रहा है आपको? पहले मुझे ये बताओ कंट्रोल सैंपल का कलर क्या होगा और ट्यूमर सैंपल का कलर क्या होगा, एज वेल एज जो पूल्ड सैंपल है उसका कलर क्या होगा.
The control should be green.
क्या कंट्रोल शुड बी ग्रीन एंड द पूल्ड इज
C2 should be yellow.
C2 is not yellow, C2 is blue and C5 is
Red.
रेड. सो कंट्रोल सैंपल के लेन्स कौन से हैं? ओनली कंट्रोल सैंपल जिनमें दिख रहा
1 and 6. 1 and 6 is control.
है. हाउ? Control is labeled with,
Oh, oh, green. It should be
yeah.
green. So two,
So
8.
2 and 8, yeah. 2 and 8. Fine. So answer is 2 and 8. Then which dye are depicted in lane 5 and 10 of the gel images from the above question is are utilized to label the sample. So dye which is present in 5 and 10, वो किसका additive effect होगा? Because definitely it's an additive effect. So lane 5 में क्या है और lane 10 में क्या है? Think and answer. I think it's very easy. If you know the fluorescence, if you know their emission, their fluorescent color, then you know.
Ma'am, I think it's C.
C. So you're saying C by 2, C by 3, C by 5. Any other answer? Answer here is C. True. Correct. How 5 में कैसे पता चला कि green B है? क्योंकि रेड तो काफी क्लियरली दिख रहा है, रेड एंड ब्लू, बिकॉज दैट्स व्हाई इट्स गेटिंग पर्पल. बट ग्रीन भी है, ये देखना था, यही इसका कैच है, because you have a green effect here in this particular region. See, right? The blue is getting shifted towards the lighter blues. That is because of green. So all the three are present, and or I would say it's turning a little white. सो तीन का इफेक्ट वाइट ही है, बट बिकॉज ये पर्टिकुलर प्रोटीन तीनों में प्रेजेंट होगा, दैट्स वाई ये वाला बैंड थोड़ा सा वाइट है, बाकी ये वाला पर्पल है. दैट मीन्स ये रेड और ये रेड, ये बेसिकली आपके ट्यूमर सैंपल में ज्यादा प्रेजेंट है ये वाला प्रोटीन. सो दैट्स हाउ यू गेट टू नो दिस थिंग. सो आंसर हियर इज सी. Next question is, which is the in degree and out degree of the red and yellow nodes in the given below network? So sir might have talked about the network, how you get the interactome, or basically how you make the protein network. So there's an in degree and there's an out degree. So in degree is basically from where that particular network is coming. Out degree is where it's going. So you have to tell red ka in and out, yellow ka in and out. In is
Uh,
from where it's coming, and out is where it's going.
Ma'am, is it B?
B, no.
Oh, okay. I mixed up the colors.
Yeah. So for red, it
It
is
it
1.
should be
Hmm. It should be
A.
A, yeah. So for red, it's coming from this particular network and it's going nowhere. Like it's not leading to any other network further. But for yellow, it's coming from this particular node and it's giving rise to 3 nodes. So it's 1, 0 here. Yeah, Hari, you can ask.
Ma'am, I have this question regarding these networks, so these graphs. So, like with biological systems, are all networks that we make directed? Because Sir was explaining about directed and undirected graphs.
Hmm.
So, like when we talk about like biochemistry or like proteomics
Hmm.
specifically, do we have like undirected graphs?
So
Like
for proteomics, I think it's yeah. Uh, so for proteomics, sometimes you have undirected, but for transcriptomics and other genomics based studies, you have mostly directed.
uh, Like, can you give me an example of the undirected one, ma'am?
So can you tell me what do you understand by the directed one first?
Directed one, like for example,
One, yeah,
glycolysis. Like you have
right.
one step leading to another.
Another, right. Likewise, so for undirected one, because what happened when you talk about the protein interactions? What you understand by protein interaction? So basically, when I'm talking about proteomics, think it like that way. So for example, if you talk about transcriptomics, one transcription leading to expression for another transcription or down regulating another transcription, right? One phosphorylation. In case of proteomics also, you have sometimes you have directed, but mostly you will get undirected networks as well, because there is no direction to some protein is so for example, one protein is actually involved in autophagy, but that protein is also helping in trafficking, or that protein is also is a receptor of some other signal. So so there is no direction as such there.
Okay.
So one of the very good example, if I give you, I think when you have an, not exactly, how would I tell this? Let me just reframe this thing and I'll get back to you on this. But definitely proteomics can be indirected one.
Thank you, ma'am.
I'll give you the correct right example for that. Okay. Should I move forward to the next question?
Yeah, ma'am.
Okay. So this one is here. Okay. So three networks are given, are listed. Find the red nodes individual clustering coefficient for each network and arrange the network on the basis of their clusteral coefficient. Have you done that cluster coefficient? सर ने पढ़ाया है ये, how to get the cluster coefficient. Actually मैंने यहाँ पे डाला नहीं, but I was expecting that you guys are aware.
Ma'am, is it C?
How? C is no. C is not the answer.
C will have the most amount of. Oh, sorry. C will have the highest amount of
Hmm.
clustering coefficient,
Yeah.
followed by B, and I think A will have the least amount of clustering coefficient.
Are you sure? So
I'm not sure about A or
you
B,
have
ma'am.
to check the red node. Find the red node's individual clustering coefficient. Node आपकी red है. So in case of A, red is not at all associated with. Uh, I mean, it's not associated with the third one, right? So A सबसे कम नहीं होगा.
Yeah, yes, ma'am.
And in case of B and C, sorry, I think I said something, मैंने उल्टा बोल दिया. B और C में एक बार देखो इसको.
डार्क स्पॉट्स बार बार वापस आए, विटामिन सी के पावर को दुगना करो. न्यू गार्नियर विटामिन सी डे एंड नाइट सीरम. इन द डे यूज आवर नंबर 1 विटामिन सी सीरम जो डार्क स्पॉट्स को कम करे. एट नाइट यूज 10% विटामिन सी सीरम जो डार्क स्पॉट्स को सीमित रखे. डार्क स्पॉट्स को कम करे 2 times faster. न्यू गार्नियर विटामिन सी डे एंड नाइट सीरम बाय गार्नियर naturally.
So that's all I'd like to say about path lengths for now. Next, I want to focus for a little while on the notion of a clustering coefficient. Now, you might be less familiar with this notion of clustering coefficient than you are with paths and graphs. So let's go through how this is often defined. What the clustering coefficient is trying to capture is, as I said before, cliquishness. How likely is it that two nodes that are connected are part of some kind of larger, highly connected group of nodes? So one typical way of defining the clustering coefficient is to start off by talking about the clustering coefficient for a particular node in a graph. And we're going to generalize this to clustering coefficients for the entire graph next, but let's focus on a single node. So let's say V is that node, and K sub V is its degree, and N sub V is the number of links between the neighbors of V. So let's look at this example graph to ground out some of these concepts. So here we've got a graph. Let's call this node V. You could point to any of them, but I'm going to go with this one. Let's take a look at the neighbors of V. Those are the nodes that are directly connected to V in the graph. We've got this one, and this one, and this one, and this one. So the number of those neighbors is exactly the degree of V, which in this case is 4. And now let's look at the links between these neighbors of V. So there's this one that goes between the two pink nodes, and that seems to be it. This node isn't connected to any other pink node. This one's not connected to any other pink node. This is not connected to any pink node, and so on. So in this case, NV is 1. Now we're going to combine these quantities together as follows. We take twice the number of neighbor links divided by the degree of V times 1 minus the degree. So the number of those neighbors is exactly the degree of V, which in this case is 4. And now let's look at the links between these neighbors of V. So there's this one that goes between the two pink nodes, and that seems to be it. This node isn't connected to any other pink node. This one's not connected to any other pink node. This is not connected to any pink node, and so on. So in this case, NV is 1. Now we're going to combine these quantities together as follows. We take twice the number of neighbor links divided by the degree of V times 1 minus the degree of V. Now this may look a little odd, but let's compute what it is, and then I'll explain it. So in this case, 2 times NV, which is 1, divided by K times K minus 1. So that's 4 times 3. And the result we get in this case for this particular V is 1/6. So what this
Answer. I'm looking for the answer. You guys are solving or not? Okay, fine. Let's see. A से start करें अगर A में कितने neighboring, I mean red nodes में कितने neighboring links हैं?
2.
Two. Two. And how many nodes they have? Like how many links between the neighbors? So NV जो हम निकालते हैं. Link between A में. A में link है neighbors के बीच में?
Yeah.
Yes. So यहाँ पर एक link है. Right? So the coefficient going to be? So you have two neighbors. So your K value is 2. You know the formula, right? The coefficient is to the number of links between the neighbors upon में its degree kv k degree minus 1 मतलब degree into degree minus 1. This is the formula, right? I have
Uh,
not given you the formula here, but are you aware of the formula?
no, ma'am.
No, you're not. Okay. So, how I'm going to show you. Give me a minute then. I'll paste it here somewhere. Just give me a minute. I'll paste the formula here for. Can you see the screen? Can you see it? Is it visible to you? The
Yes,
formula.
ma'am.
Yeah. So what happened here is, wait a minute. Okay, it's not very clear picture, but I think you can see that. So clustering for coefficient is the number of link between all the members of neighborhood divided by the total number of link that could be possibly exist given the number of nodes. So links भी हैं इसके अंदर. So here if you could see here, okay, wait a minute. नीचे करते हैं. So in this case, the degree, that means the k is all the nodes, all the neighbors of this particular node. So it has two neighbors, right? So that is 2, right? In case of A, ki is 2. In case of k, in case of A, the ni is 1 because उसके neighbors के बीच में एक link है. So the answer here going to be 2 into 1, oh, sorry, 2 into, yeah, 2 into 1 upon me 2 into 1. That gives 1, right? So for B, this is for A. For B, how many links, how many neighbors is particular nodes के? This particular node. There are 3
Two.
neighbors.
Oh, yeah, three neighbors.
So K value is 3. And is there any link between these neighbors?
No, it should be zero, no?
Yeah. So it will be, there is no possible link. There is no possible direct link. डायरेक्ट लिंक देखना बहुत जरूरी है इसमें बिटवीन द नेबर्स. सो
Ma'am,
डायरेक्ट लिंक यहाँ पे कितने हैं? है डायरेक्ट लिंक, एक ये है और एक ये है, ये वाले कैंसिल्स कर रहे हैं हम, दैट्स व्हाई हमने सिर्फ एक ये डायरेक्ट लिंक लिया इस केस में, बिकॉज दीज आर द नेबर्स, ये बिकॉज ये नेबर नहीं है उसका, बट हियर देयर इज अ लिंक बिटवीन दिस एंड देयर इज अ लिंक बिटवीन दिस। सो कितने
So
लिंक हुए?
two links, man.
टू लिंक्स, सो टू लिंक्स हुए, सो 2 into 2 अपॉन में 3 into 2 कितना हुआ? 1 by 2 by 3 which is less than 1, right? In case of C कितने K हैं हमारे? 3, एंड लिंक्स कितने हैं? 1,
Uh,
2
links also
एंड
should
3,
be. Yeah.
3 ही लिंक्स हैं और 3 ही हमारे वो हैं in case of C, so the coefficient is 3 into 2 upon में 3 into 2, that is also 1, so
Yeah.
answer here going to be D। समझ आया ये चीज?
हाँ man.
अभी
Yeah.
आ गया समझ? ठीक है, तो ये फॉर्मूला आपको इसके साथ लगाना है, सो आंसर गोइंग टू बी डी। Next question quickly. Three different types of network are typically investigated in system biology. Which of the following, when applied to these networks using graph theory, is correct for the general classification? Protein and metabolite network undirected होते हैं, transcriptional network directed होते हैं. Protein network undirected, transcriptional and metabolic networks directed. Protein and transcriptional network directed, metabolite network undirected. Protein network directed, transcription and metabolite network undirected. I have already given you the answer of this question. Quickly, I want the answer.
Should be C.
It's, yeah, it's what?
C. C.
D.
C, man. C.
C. C for cat.
Yes.
How? I just told you that protein is undirected. While your transcription, because as you mentioned, glycolysis, it's a directed one. Transcriptional, it's a directed one. The protein networks are generally undirected. There might be interaction between two unrelated proteins. Unrelated as in वो भी नहीं है, network में भी नहीं है, signaling में भी नहीं है, but they are interacting in a different process. So proteins are undirected, transcriptional and metabolic networks are directed. Quickly to the second last question. Three gel pictures. I don't know whether it's visible. It's visible, I guess. From a single proteomic experiments are provided here. Which method from the list below would have been used to obtain gel image like this? 3 2D gel stained with fast screen, Propu diamond and Kumasi stain. 3 I-track labeled sample followed by 2DE and staining. 3 two-dimensional gel stained with fast green, Nile red stain and Kumasi blue. And DIG labeled sample followed by two-dimensional experiment and scanning at a different wavelength. It's a very simple answer. तो ये तीन इमेज हमारे पास कैसे जनरेट हुई?
Oh, D.
Yeah. So we have done differential in gel electrophoresis where we have used three different dyes. So answer here is D. Last question. Given below are the few steps involving during a gel-based proteomic experiment. So there are step 1 for CY labeling, step 2 sample pooling, 3 isoelectric focusing, 4 SDS page, 5 dehydration of IPG strips, 6 internal standard, 7 gel staining. So you have to tell which of the following pair best describe gel-based proteomics technique and associated steps. सो वन डायमेंशनल के साथ कौन से स्टेप्स हैं, टू डायमेंशनल के साथ कौन से स्टेप हैं, और टू डायमेंशनल डिफरेंशियली इन जेल इलेक्ट्रोफोरेसिस के साथ कौन से स्टेप्स हैं. वेरी सिंपल क्वेश्चन वैसे तो. टू डी में कौन से होंगे? we do 1D में हम क्या करते हैं? 1D में क्या रन होता है?
Ma'am, is the answer C?
Okay, so 1D में you are saying that you have, okay, then 2D में 3 4 5 7, okay, then 3D में, okay, let's see the answer. So answer is C, you're right. 1D में we have SDS page and gel staining. 2D में we have sample, isoelectric focusing, SDS page, rehydration of IPG strip and gel staining. And in case of 2D DIGE, we have 1 to 6 steps, right? So that's all from my side. Any other question you have? Okay, then thank you for joining, and I'll meet you in the on next Tuesday. And please do watch all your videos before joining these live session. That will help you a lot. So for example, I thought that you are aware of coefficient, uh, this clustering coefficient. You are aware of the formula. So next time I want you to guys to go through. That's why I have not given you, or I have not taught about this, because it will take a lot of time and we just have 1 hour for teaching and 1 hour for the question answer. But still I went 1.5 hour for the teaching thing. So that's why it is actually a very less time for me to cover all the concepts. But I want you guys to watch those videos so that you can relate to this quick live session. Okay, anything, Hari? Do you want to say anything?
No, ma'am.
Okay, fine. Itishri, anything from your side? Okay, cool. I thought you guys are saying something. Thank you so much for joining. See you next week.
Thank you.
Sorry, I could not hear
Thank you,
you,
ma'am.
Itishri.
Thank you, ma'am.
Thank you so much. Thank you. Okay, fine. Thank you so much for joining.